In this video, we will discuss about Ca2 plus buffering function of Calreticulin. So, we will continue our discussion where we have stopped in the last video. And in this, the main role in Ca2 plus buffering that happens is between the endoplasmic reticulum and the protein present in the plasma membrane. So, as we know from our previous videos that calreticulum is responsible for around 50% of the calcium that are available in ER lumen, and there are other Ca2 plus binding protein also in the endoplasmic reticulum. So how this Ca2 plus release in the endoplasmic reticulum with the help of receptors. One is uh, inositol 145 triphosphate receptor, and another one is rhodine receptor. So whenever there is a fluctuation in the Ca2 plus level in within the endoplasmic reticulum or within the cell, these receptors get activated, and that helps in release of the signal for that fluctuation reaches to the endoplasmic reticulum, and then it further reaches to this, further activates or directs this receptor to release Ca2 plus from the calreticulum and the other proteins that have Ca2 plus within the endoplasmic reticulum. So once the Ca2 plus is released, it is essential to refill that Ca2 plus uh, for further future events. So that thing has been done by Sarka process. We will discuss about Sarka process in another video. But what happens in this Sarka process is that uh, there is a signal that is being sent to the plasma membrane from where the additional Ca2 plus requirement of the requirement of Ca2 plus in the endoplasmic reticulum is been fulfilled, and this figure shows. Let me open the laser pointer. Yeah, and this figure shows the C domain of the calreticulum, uh, which has in this C domain around most of the amino acids are acidic, and out of last 82 amino acids, 43 amino acids place uh, plays a role in the Ca2 plus binding or Ca2 plus buffering. And this figure shows here, this one is the nucleus, and this one is the endoplasmic reticulum, and this one is the plasma membrane. So there is a signal, or you can say talk cross. There is a talk between endoplasmic reticulum and the plasma membrane during Ca2 plus cycle or the Sarka cycle, and uh, inefficient level of Ca2 plus within the endoplasmic reticulum may affect the endoplasmic reticulum Golgi apparatus trafficking. That is the further that is the transfer of proteins from endoplasmic reticulum to Golgi gets affected. At the same time, it will also leads to ER stress that result in regulation of translation of proteins, and at the same time also upregulation of chaperones and ERED uh, and ERED proteins that are that take part in the ERED, because in whenever the Ca2 plus levels are decreased, the signal that cell receives is that there is some kind of problem with the uh, functioning of the endoplasmic reticulum, and one of the main function of the endoplasmic reticulum is to provide or to help proteins to acquire their native structure. So this Ca2 plus release so this Ca2 plus level fluctuation maybe is one of the indication that uh, is one of the indication that that the functioning of endoplasmic reticulum is not normal. And in the previous slide, we have discussed that Ca2 plus binding occurs in the C domain. However, in P domain, that is protein rich domain, there is also Ca2 plus binding happens. What is the difference in between those binding is that in C domain, the binding is of high capacity and low affinity. That is as we discussed about this numerically in our previous video, that per mole of this calreticulum or the protein can bind around 25 mole of Ca2 plus. However, in case of this protein rich domain, the capacity has been reduced to like 2 mole of calcium per mole of proteins. The value is not exactly correct, but it is somewhere around this. And the affinity is also been increased. Here the affinity is around 2 nanomolar. Here the affinity might increase to around 10 nanomolar. So the release of Ca2 plus is much easier in case of C domain than in case of P domain. And these are some of the proteins that are responsible for maintaining Ca2 plus homeostasis. So when the Ca2 plus is released in the endoplasmic reticulum, that is here, there is a protein available at the endoplasmic reticulum membrane, that is STIM1, that signals the Ca2, that measure, or that is the sensor for measuring the Ca2 plus level, or measuring the Ca2 plus level within the endoplasmic reticulum, and gives a signal to the protein data available in plasma membrane, that is SOCI protein, store operated calcium influx protein. And, and it is not directly gives signal to the SOCI. However, it gives signal to this ORI, which is the Ca2 plus transporter. So it is also present at the plasma membrane. So it gives a signal to ORI. Then this can direct SOCI to arrange Ca2 plus from the extracellular matrix, and from the plasma it brings the proteins from the extracellular matrix towards close to the plasma membrane. And from plasma, intracellular transport has been taken will be taken care of by ORI1 to transport the Ca2 plus from plasma membrane to the endoplasmic reticulum. So this is in brief how the Ca2 plus homeostasis is maintained within the animal cell. So thank you for your time. Have a good day.